Today we will discuss about the in vitro migration assay overview. So this is not a topic which is specific to a particular area, but we will understand how the in vitro migration assays evolved with time and what are the bases for this in vitro migration assays and which of them are most commonly used. So first we will start with this basis. So what is the hypothesis generally in the migration assay is that these are the cancer cells when they interact with their ECM or when they change the surrounding matrix, and with time what will happen is that as the cancer progresses there is an increase in the stiffness in the ECM surrounding the cancer cells, and which are commonly known as tumor microenvironment. And with increase in ECM, the stiffness with increase in ECM stiffness, so there is a physical interactions occurs between the ECM and the cell surface receptors that will trigger more cancer progression properties such as proliferations and migrations. So generally what people are expecting is that the with increase in stiffness the migration and metastasis properties of the cancers should increase. And what are the different migration assays that can be carried out in vitro in the lab? So these are mainly categorized into 2D culture and 3D culture. In 3D cultures, these are three different categories that are shown here. First one is this MCT and tissue-derived scaffold and synthetic scaffolds. Out of this, the MCTs have this low cost and reproducibility, but the difficulty is that they are poorly regulated by physical properties. The interactions of the cells with the extracellular matrix is not that good. And whereas in the case of tissue derived scaffold, their reproducibility is the concern, but they provide better cell adhesions and biocompatibility. And when it comes to the synthetic scaffolds, they are stables, and one can easily alter their stiffness to observe the cells behavior. But what they lack is that they lack the adhesion ligands, or we can, in other terms, the ligands that actually, the interactions that actually occurs in vivo within the body or within the patients. So this is the general overview about the 3D cultures. In the case of 2D cultures, uh, these are currently widely used migration assays, where the advantage of it is that it is of low cost, easy to operate, and since most of the peoples are used to the 2D cultures, it can easily be confirmed. The results can easily be confirmed from different labs by comparing the results of the different lab. And the disadvantage is that cells are forced to be flat and elongate, which is not the case in vivo, or actually in the patients, and it lacks the proper physical stimulus. So since it does not provide a 3D environment, the 2D environment always to some extent lacks the physical stimulus that the actual cells observe when it is surrounded by the tumor microenvironment from all the directions. And sometimes because of the difference, its 2D because of its 2D culture, it will give different gene expressions compared to what has been identified in the patients who are affected with the similar kind of disease. And sometimes it provides extra high stiffness compared to what the hydrogels has been designed theoretically. So these are some of the advantages and disadvantages of the 2D culture. And Now let us also quickly go through the physical stiffness that has been identified at different tissues. In the case of lung, physical stiffness of normal healthy lungs is around 150 to 200 Pascal, whereas in the pathological case it can go up to 15 kilopascal. And similarly, there is a difference that has been shown here, and one such difference is about this pancreas, which has a physical stiffness of around 1 to 2.9 kilopascal, but uh, this pathological stiffness is around 3.7 kilopascals, and this is one of the most aggressive cancers which can proliferate and migrate with this increase in the stiffness from 2.9 to 3.7 kilopascals. So this also shows that with increase in the stiffness there is a increase in the cancer progression probabilities. So I hope you understood the brief overview of in vitro migration assays. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. All these all these two figures and one tables are from the same paper, same review paper. Thank you for your time.